So, hello students, good morning. So, in last lecture we complete first topic, that is growth, and now from this lecture we were starting second topic, that is effect of environmental factors on the growth of bacterial growth or growth of bacteria. Under this topic we are going to study different environmental factors like temperature, pH, salt, heavy metal and radiation, and what is that effect of these different factors on the prokaryotic cell, that is bacterial cell. Here this syllabus is comes under the paper of physiology. Growth and effect comes under the studies like of the physiology. Now what is these factors and how they are going to affect bacterial growth? Here, let us start from the first environmental factor, and that is effect of temperature. Temperature is most crucial environmental factor. It is most important environmental factor which is affecting the growth of all different types of organism. As you know, when we are cultivating bacteria under the in our laboratory, we must require a proper temperature for the incubation. Yes. So how this temperature was decided? It is the effect of different temperature on the growth of organism. So let us see the effect of temperature on the growth of organism. Here temperature, mm, if we grow organism at different temperatures, suppose we have a culture, and if we cultivate this same culture at different temperature, from suppose we incubate one plate at 0, then 10 degrees Celsius, 20 degrees Celsius, 30, like that. For every 10 degrees Celsius, if we incubate the culture, and if we observe the growth pattern, it was observed that at the 0 there may not be growth, but rising 10 degrees Celsius may rises the temperature. And at the specific time, up to the specific temperature, we will get the rising growth. Beyond that specific temperature, again organism lowers their growth rate. So here, suppose this is the graph. Here, from 0 we are increasing the temperature and we are measuring the growth rate. At the 0 or at the low temperature, there is low growth or minimum growth. As temperature rises from 10, 20, 30, like that, for every 10 degree rise in the temperature, increases or doubles the growth rate up to certain limit. Suppose this is a temperature. You may observe this temperature. Here we will get the highest growth rate. Yes. Now, after that, what happens? After that, increase in the temperature lowers the growth rate. Now, why this happens? This is the actual effect of temperature. Rise in temperature up to a certain limit increases growth rate. Beyond that temperature, increase in the temperature decreases the growth rate. Here, while studying this temperature, effect of temperature, we will get three different ranges or three different values. This is the lowest temperature, below which if we decreases more, if we decreases the temperature beyond this value, there is no growth. So this is the minimum temperature. Minimum is the temperature below which organism cannot grow. Then this is the another extreme end. Here at this end, if we increase the temperature beyond this temperature, beyond this value, if we further increase the temperature, again organisms will not grow. So this is the maximum. Maximum is the highest temperature above which organism cannot grow. Minimum is the lowest temperature. Suppose it is the 0. Below 0, organism will not grow. Suppose it is 80. Above 80, organism will not grow. So these are the extreme values, minimum and maximum. Within minimum and maximum temperature range, organism shows the growth. Now what is this? It is the optimum temperature at which organism shows highest growth rate. Suppose we draw this line on the y-axis, we will get highest value of the growth rate. So optimum is the temperature at which organism grows with the highest growth rate. And all of you know, we are cultivating organisms, we are incubating organisms at the optimum temperature. So our incubator is adjusted to the optimum temperature. Generally, we are using 37 as the optimum temperature for our all routine bacteria. So this is the optimum temperature, and this minimum, optimum and maximum temperature, this range of the temperature in which organism shows the growth is called as cardinal growth temperature range, or these are the cardinal growth temperature values, and they are specific for specific organism. Every organism has its own cardinal growth temperature range. For example, some organisms start their growth from 15 and ends up to the 80 degrees Celsius. So their minimum is 15, 80 is their maximum. Another example is, suppose the organism starts its growth from 0 and can able to grow at the 40 degrees Celsius. So it's 0 is the minimum, 40 is the maximum. So this is the cardinal growth temperature range, or this is the temperature range required by the each organism. So that is the first point. So here let us revise. Temperature increase the enzyme activity, so it increases the growth rate up to the optimum temperature. Too high temperature, so this is too high, beyond maximum. Too high temperature diminish enzyme activity. What is diminish? Diminish means inactivate enzyme activity or stops enzyme activity. It denature the protein. It denature, it disturb the denature. The meaning of denature, it destroy the structure of proteins, and all of you know proteins are our enzymes. So enzymes get inactivated. Ultimately, organism cannot able to grow. Every bacterial species has its own specific growth temperature. As I told, optimum is the temperature required for the growth of organism. Every organism has its own growth temperature. Here cardinal temperature values, that is minimum, optimum, maximum. These are the cardinal growth temperature values within which organism shows its growth rate. So that is the first part under the effect of growth. Now, what is the actual effect of growth? What happens? Why there is no growth below minimum temperature, and why there is no growth above maximum temperature? Now, what happens? The change in temperature beyond required growth temperature. Beyond means below minimum and above maximum. Beyond required growth temperature range affect the in opposing ways. What is that opposing ways? At low temperature, there is different effect. At too high temperature, at very high temperature, there is another effect. Now, at the lower temperature, let us see what happens at the lower temperature. At lower temperature, usually increase in the metabolic activity, increase in the, as there is increase in the temperature. From 0, as I told, if we incubate organism at 0, that is low temperature, metabolic activities are very slow or 0, but as we increase the temperature, metabolic activity will increase. Now, this is due to increase in the rate of enzyme catalyzed reactions, causing overall increase in the growth rate. Now, why this increase happens? This is due to the increase in enzyme activity. Now, as I told, usually rate doubles for every 10 degrees Celsius rise. Hmm? So, growth rate doubles for every 10 degrees Celsius. For example, 0, 10, 20, 30, like that. For every 10 degrees Celsius rise, growth rate doubles. After a certain limit, rise in the temperature lowers the growth rate. As I told, up to the optimum, growth rate was double, but beyond optimum, growth rate again decreases. Why growth rate decreases? Because of thermolabile nature of the enzyme and all cell components. Now, what is thermolabile? Thermolabile is nothing but labile is sensitive. Thermo means temperature. Organism or cell has thermosensitive, temperature sensitive enzymes, and all cell components are there which may get denatured due to the high temperature. And because of that, beyond certain limit, rise in the temperature lowers the growth rate. Now, what is the effect of high temperature? Suppose, as an example, suppose organism has a growth temperature range from 0, sorry, from 15 to 45. 15 is its minimum. 45, for example, not 45 actually, it is 65 or 70. 15 to 70. Suppose organism has minimum 15 and maximum of 70 degrees Celsius. Now what happens beyond 70? If we raises the temperature to 80, 90 or 100 degrees Celsius, what is the effect of this high temperature? Here the effect of high temperature is permanent and irreversible. Irreversible means we cannot regain bacteria. If we treat organism at 100 degrees Celsius, permanently bacteria will kill. We cannot obtain, we cannot regain, we cannot return this bacteria towards their normal cells. So this is a permanent and irreversible effect. Now what happens at high temperature? At high temperature, it changes the rate of biochemical reaction. It causes the denaturation of enzyme. It causes the damage to transport carriers. As I told, denaturation means destroying the structure of enzyme. It high temperature cause damage to transport carriers. Carrier proteins get damaged. Then destruction in the cell membrane. Cell membrane, our cell membrane is very sensitive. It get destroyed due to the high temperature. Then next is it changes the composition of lipid. High temperature cause changes in composition of lipids. Now where lipids are there? Lipids are especially present in our cell membrane. At low temperature, higher amounts of unsaturated fatty acids are found, and at very high temperature, saturated fatty acids are largely found in the cell membrane. So that is the change occur at the high temperature. Now, due to the very high temperature, pigment production ability was slower. There is low pigment production. Certain bacteria have property to produce colored pigment, but due to the high temperature, that production of pigment was slower. Then next is there is melting and disintegration of liquid lipid bilayer, not liquid, it is actually lipid bilayer that leads to loss of structural integrity and loss of fluidity of the membrane. So that is the major effect. It causes melting and disintegration of lipid bilayer of the cell membrane, which affect our property of the fluidity. Cell membrane is fluidic, and that fluidity was lost due to the high temperature. The next is last one is growth is inhibited because of non-repairable cell damage. Once cell get damaged due to the high temperature, these damages cannot be repaired again. So that effect is irreversible effect. So that is about the effect of actual high temperature. Now what happens at very low temperature? Suppose we decrease temperature below minimum temperature. As I told, below minimum, again organism cannot grow. Now effect of temperature is at the low temperature, it is temporary and reversible at the low temperature. For example, we are preserving our culture in the freeze. Now what happens at the freeze temperature? Freeze temperature is low temperature, but at the freeze temperature organisms cannot grow, but they are not killed. They are just inhibited to grow. Inhibited to grow, they are not allowed to grow. But if we are taken out that cultures from the freeze, and if we expose same culture to the room temperature, they start their growth again. So this effect is not permanent. Effect of low temperature is temporary effect, only for a time. When we are storing culture in the freeze, for that time only that culture may shows growth inhibition. After returning that culture at the room temperature, organism starts the growth. So that effect is temporary. Now cell may be permanently affected at the low temperature. Now when this low temperature permanently kill bacteria, for example, suppose we preserve culture in the ice box, freezer, where temperature is zero, near about zero. At the zero temperature, all of you know what happens. At the zero temperature, ice formation is there. And suppose when in such case, if we preserve the culture at 0 degrees Celsius, then there is ice formation. And once there is ice formation, that may cause the mechanical injury to the cell. cytoplasmic water content is converted to the ice, and because of that ice crystals in the cytoplasm, cell membrane get damaged and cell was killed. So at that freeze temperature below zero, the temperature which allows ice formation, the effect of that temperature is permanent effect. So at freezing temperature, ice crystal formation of cytoplasmic fluid occur, which may mechanically damage and rupture the cell. In this case only, low temperature permanently damage the cell. Otherwise, the temperature just above freezing, that is called a chilled temperature, cause the temporary effect. Now low temperature above freezing, that that I said as a chilled temperature, just above freezing. We are not allowing ice formation. That the temperature might be 4 degrees Celsius or near about 4 degrees Celsius, where temperature is low but there is no ice formation. It is called a chilled temperature. What happens at the chilled temperature? Here there is prevention or restriction of the solute transport. At this chill temperature, organisms are not growing properly. They stops their growth for a while. For a time when they are at that same temperature, they cannot able to grow. They inhibit their growth. They stops their growth temporarily. Here, because there is prevention or restriction of solute transport, there reduced enzyme activity, reduced ATP production, pigment production, flagella formation is also reduced. Here there is synthesis of intracellular, extracellular polysaccharides also affected. Because of these different effects, bacteria cannot grow. But if you shift that culture to room temperature, immediately organisms start to grow by taking control over all these effects. So chilled temperature gives the temporary effect, but ice formation or freezing gives the permanent killing in case of the cell. So these are the two different effects of the low temperature. Now here there are number of different types of organism depending upon the temperature requirement. As I told, every organism has its own temperature, cardinal growth temperature range. Suppose we have two example. One organism cardinal growth temperature from 15 to 70. Hmm. And another organism has cardinal temperature from 0 to 25. In those two cases, this cardinal growth temperature range is changed. And depending upon this temperature, some organism has very low temperatures as their cardinal growth temperature range. Some organism can grow at very high temperature and some will grow at the middle temperature. Depending upon that, there are different physiological groups of the organism. Depending upon temperature requirement, organisms are divided into different types. One is first is mesophilic. Meso means middle. Philes means requiring or loving. Organisms which are loving moderate temperature or organisms which are requiring middle range of the temperature. So which require middle temperature range for the growth. Then next is what temperature range they requires. Their temperature range is 25 to 40 degrees Celsius. There is the optimum. Their minimum temperature is 15 to 20 and their maximum is 45. So their temperature range starts from 50, 15, that is their minimum. Their optimum is 25 and their maximum is 45. So these mesophiles can grow between 15 to 45 degrees Celsius. And examples are most of the common spoilage causing organism as well as our pathogens, that is disease causing organisms, are comes and will come under the group called mesophiles. They all require the middle range, that is 15 to 45. It may extends up to the 60 or 65 degrees Celsius, beyond above which they cannot grow. Here organisms that have adapted to live in the animal body, that is disease causing organisms like Salmonella, E. coli, all our routine bacteria which we are using for practical comes under this group Mesophile. So all pathogens and common spoilage causing organism. Spoilage means spoilage of the food. Food spoilage causing organism comes under the group Mesophiles. Now which is the second group? Here second is Cyclophilic. Cycros means low. Philic means requiring. Organisms requiring cold temperature for the growth. Here low temperature requiring organism. Here there are two types of the Cyclophile. One is obligate Cyclophilic and another is facultative Cyclophile. Obligate means they must require low temperature. Otherwise, they will not grow. Facultative means they can grow just above the low temperature. Now, which are the temperature requirements of obligate saprophiles? This is optimum temperature is 15, minimum is 0, maximum is 20. So, temperature range is 0 to 20. 0 is minimum, 20 is maximum. Within this range, organism will grow. Hmm? They are very sensitive to the warm temperature. That's why they are called as obligate. Obligate means strict. They strictly require very low temperature. Just a warm temperature cannot allow their growth. So they are very sensitive to the warm temperature, cannot even tolerate the temperature equal to 25. 25 is also cannot allow the growth of obligate saprophiles. Now, which are the examples? Examples are organisms residing in deep ocean, Arctic, Antarctic habitats, polar regions, etc. And another is facultative means which can grow just above the low temperature. Here these organisms grow at 0. They have their optimum temperature 15, maximum about 35 degrees Celsius are called as cycrotrophs or they are called as facultative cyclophilic. Temperature ranges 0 to 35. Up to the 35 they can grow. Here organisms causing spoilage of food preserved in refrigerator. We are generally observing that suppose we kept a food in the refrigerator, especially any milk product, if it was kept in the refrigerator for a prolonged time, then it may get spoiled. So these organisms which cause spoilage of food stored in refrigerator comes under this group. And examples are alkaligenes, Flavobacterium, Serratia, Candida, etc. And last one is Cyclophilic, can grow at low temperature. Now why these cycrophiles can grow at the low temperature? Because their enzymes transport system, that is transport carrier proteins, then protein synthetic machinery, that is ribosomes, functions well at low temperature. Their cell membrane contain high unsaturated lipids which may remain semi-fluid at the low temperature, and that is how they get maintained at the low temperature. So that is the second type, that is cyclophilic. And now last type is thermophiles. Thermophiles are heat-loving organisms. Their optimum temperature is 55 to 60 C. Their optimum is very large, high temperature at 55, 65 degrees Celsius. They will show their maximum growth. Their minimum is 25 and their maximum is 70, or it may extend up to 100. So 25 to 70 or 80 is the temperature range. Many unable to grow at 45. Even slight decrease in 450 will not allow the growth of thermophile. Spore forming bacteria and organisms found in organic compost piles are the examples of thermophile. For example, Bacillus stereothermophilus, Thermus aquaticus, Clostridium thermocilium, etc. Now there is another group, hyperthermophiles. Hyper means again very high. Hyper means high temperature. Here temperature is 70, that we are saying as a thermophiles. Beyond 70, some organisms can grow up to 100 degree. That is hyperthermophiles. Grow above 80 as an optimum. Their optimum is 80 and their maximum is 100 and minimum is 55. Below 55 they cannot grow. At 100 they can grow. Now, where these organisms generally occur, they are found in hot springs. All of you know where, how, what is this hot springs? Hot springs, very near to Patan. When we are going to Chiplun, there is one village, Someshwar. Yes, at the Someshwar, I think all of you observed these hot springs. The some organisms resides in such hot springs. These are hyperthermophiles. Volcanic areas also shows the presence of hyperthermophiles, and these are some examples of the organisms of hyperthermophile groups. Now, they tolerate and survive. Their proteins and enzymes are more heat resistant. That's why they tolerate high temperature. Their protein synthetic machinery functions well at high temperature, and cell membrane have more saturated fatty acids. Here, these saturated fatty acids have high melting point. So, at very high temperature, generally these fatty acids get melt, but due to the this composition, saturated fatty acid, their melting point was increased, and they do not get denatured. So that is the actual reason why this special group of organism can tolerate such high pressure, high temperature. So that is about the last type of the temperature requiring organism, that is thermophile. Now there are certain terms we required to study the effect of temperature. When you are setting any temperature, when you are studying a bacteria for its temperature requirement, then we must know the some values, or we must know some terms. At the same time, suppose we are killing organism. Now where we require to kill organism, yes, we require this killing in the sterilization. So suppose you are doing sterilization. In that case, you have to kill the organism by using temperature. Secondly, if you are cultivating organism, for example, in case of fermentation, you must know the proper temperature for the growth. So for cultivation of organism, at the same time for killing of the organism. For both these cases, we must know some values. We must know some parameters. Which are these parameters? These different measures include TDP, that is thermal dead point, TDT, that is thermal dead time, and D value. Now what is this TDP, thermal death point? This is the lowest temperature at which organisms from a culture get killed within 10 minutes. Suppose you have culture in the test tube. Within 10 minutes, you have to kill that organisms, all organisms, and the lowest temperature required for this reason is called as thermal death point. Hmm? Now, thermal death time is the minimum time. It is the shortest time that is required to kill all bacteria in the specified temperature. In this second case, we are using specific temperature, suppose 37. What should be the time required to kill organisms at 37? That time is called as TDT. And D value is the, in these cases, we are saying that organisms time required to kill, temperature required to kill all organism, and here it is the time required to kill all organism, but it is impossible to kill 100% organism, and that's how this term D value comes. D value is the decimal reduction time. It is the time in minute at a given temperature required to destroy 90% of the targeted organism. Suppose we have culture, then from that culture 90% population should get killed at a given temperature at a given time. That is called as D value. So we must know this D value, TDP and TDT. So that is about the effect of temperature. The next effect is effect of pH, that we will see in the next lecture. For this time we stop here.